Every child with cancer is unique.
Every child's cancer is different.
But in each cancer, there is a weakness.
We identify that weakness and look for a drug that targets that cancer.
Hi, I'm Dr. Chris Maxwell.
Hi, I'm Dr. Philip Lang.
Hi, I'm Dr. Gregor Reed.
Hi, I'm Dr. James Lim.
I'm a proud researcher in the
proud researcher,
Cuccione childhood
proud
cancer
member
research
of the
program.
Michael
I'm a proud
Cuccione
researcher
Childhood
of the
Cancer
Michael
Research.
Cuccione Childhood Cancer Research Program.
At BC Children's Hospital, about 130 kids a year are diagnosed with cancer. Thankfully, many and most of those kids will survive their disease, about 80 to 90%. Unfortunately, some of those kids will relapse, and the relapse survival is only around 20 to 30%. What we do in the BRAvE initiative is we grow a representation of a child's cancer. We do that so that we can find out as much information as we can about that cancer. We can look for weaknesses, and when we find a weakness, we look for drugs that target that weakness, that are matched to that weakness, and then, because we have the avatars, we test those drugs against that child's cancer. In the BRAvE initiative, my lab oversees the genetic sequencing. That is, we look for how the genes might change in a cancer with respect to normal tissues. And once we identify those changes, we pass it off to others in the BRAvE initiative to target those weaknesses.
In the BRAvE initiative, we use cutting-edge technology to weigh and count thousands of proteins and create maps of the proteome in a drop of blood or a small biopsy. If a protein is altered, a cell could begin to multiply uncontrollably and ultimately lead to cancer. Or, if another protein is altered, a treatment may fail to work or cause serious side effects.
In the BRAvE initiative, I'm sort of known as the mouse guy. It's my lab's job to take cancer cells that we are able to obtain from the biobank here at BC Children's Hospital that are isolated from children with cancer and grow those cells up in mice, immune-deficient mice, so you can grow the human cells. And what this does is it gives us a lot more cells to work with. For doing the sort of experiments that the other researchers here are doing, you need a lot of cells. So by growing them up in mice, we take what is a patient cell and we turn maybe 1 million cells into 1 billion cells in some cases. So that allows us to do a lot more research on that patient's cancer.
So for the BRAvE program, my lab tests the response of the drugs that we've identified that will target the child's cancer. So each drug will be tailored individually to that child's cancer.
It's our hope that with those matched therapies, we can better treat the relapse and refractory childhood tumors.